Well, good evening, everyone. It is my privilege to introduce to you Dr. Julia Ridpath. She is an internationally recognized expert in all things BVD. She graduated with a PhD from Iowa State in 1983 and went to work at the National Animal Disease Center in Ames, where most of her career has been spent in projects to improve the detection and prevention of BVD viruses. I asked Dr. Ridpath to put this together for beef producers specifically about the BVD virus and with special attention on PI calves. If you have any questions or you don't understand some of the words we're using, please type your question in the question and answer box. So without further ado, I'll turn it over to Dr. Ridpath.
Well, thank you so much for this invitation, and I'm so happy to be able to talk to you. In these weird COVID times, we get together however we can. And as a person who spent their life trying to prevent epidemics of disease in cattle, it's kind of weird to be living in an epidemic of people. But welcome to the weird and wild and wonderful world of viruses, because just as the COVID pandemic is confusing, BVD is confusing in the same way, and that's just characteristic of viruses, as we'll talk about a little bit later. So what I'd like to start with is kind of life in the trenches. These are the typical type of things that Michelle is getting comments from the field. We kind of made up this example. I brought a load of 100 5-weight calves at the stockyards on Thursday. They were newly weaned. We tested them. We did find out a couple of PIs and removed them in 36 to 48 hours. But now we have a bunch of questions on what happens next. So the questions you can list there, I can just read them for you. How long will it take before they see sickness? How long after infection before you have an effect on the immune system of the calf? And how long will the immunosuppression last? What causes the difference in severity of the diseases we're seeing? When can one expect the virus to be out of a group of calves? I mean, when can you shed light on transmission across fences? Can they touch noses, transmission in water troughs, bedding? And how do you clean and disinfect? Well, I'm not going to answer all those questions now because I need to give you a little bit of background information. But we'll come back to these questions at the end of my talk, and we will discuss on how we would answer them. But first, I need to tell you a little bit about the bug we're working with and what it does. So BVD is actually a collection of different diseases all under the same heading. We have acute infections and we have prolonged and persistent infections. And we concentrate a lot on that persistent infection because they're trying to get rid of the persistently infected calf. But to remember that there's a lot of acute infections that are accompanying the generations of that persistently infected calf. So in acute infections, we can have enteric disease that will see diarrhea. Originally, BVD was called cow cholera. We also see hemorrhagic syndrome where you have very bloody diarrhea. We have mucosal disease, which happens in PI animals and is 100% fatal. BVD is a big contributor to respiratory disease through direct interactions with other pathogens, and also because it damages the immune system and prevents the animal from clearing other pathogens, other bacteria and viruses. But it causes its biggest hit as a reproductive problem. You can get abortion and loss of pregnancy, but the gift keeps on giving is the birth of that persistently infected animal. So we have these transient infections, the acute infections, and they last from 2 weeks to, and I have a row of question marks here. And that's because it really depends on a number of factors on how long that virus is going to hang around. Just like what we're seeing with the coronavirus now. Some animals are super shedders, shed it for a long time, are sick for a long time. Some seem to get over it in 2 weeks without a problem. But the prolonged and the persistent infections are lifelong infections. Your probably most familiar with the persistent infection that results from exposure of the fetus in the first 1/3 of pregnancy. But before puberty, some heifers and bulls that are infected before puberty can get a prolonged infection of the testes and ovaries that they don't clear. So they're not persistently infected animals, but they will have reproductive problems because the virus stays in the reproductive tract. So there are 3 viruses that cause BVD, actually, 3 different species: BVD1, BVD2, and Hobie-like viruses. The Hobie-like viruses we don't worry too much about in North America. They're primarily in South America and Southeast Asia. So primarily the 2 species we're worried about are BVD1 and BVD2, which are 2 different viruses. They're kissing cousins. They are related, but they're 2 distinct forms of the virus. So which bovine viral diarrhea will your herd experience, and how long will it last? Well, when the virus gets into your herd, the disease you're going to see depends on a number of factors. It depends on the ability of the virus to cause disease, because some of these viruses are much meaner and tougher and cause much worse disease than other types and other strains of them. So some of them don't really cause much of a problem at all, and some of them will kill animals outright. The presence of other viruses and bacteria. BVD plays well with other bugs and makes everything look worse. The age of the animal seems to be that in younger animals, we see more diarrhea. Older animals, we see more respiratory disease. If an animal is pregnant, we'll see reproductive disease. We may not see clinical disease. That animal may not look like she's sick. You don't realize she ever had BVD until she gives birth to that PI calf. And also previous exposure, either by being exposed to a field strain or by vaccination. If there's some immune response there, some antibodies due to previous exposure, the virus won't cause as bad an infection, or may not even cause an infection at all. So in any outbreak or any introduction of BVD into a herd, you have all these things going on. So you don't see one uniform presentation in a herd. Well, everybody won't get sick. Everybody won't look the same way. You'll have some that get rather ill. You'll have some that may not show you anything at all. You may not know anything walk through there at all until you start having PI animals hit the ground. But just because you don't see overt clinical disease, don't think that BVD isn't causing a problem. It always nibbles away at your bottom line because it impacts on the health of that animal. So it really is the gift that keeps on giving. So when we're talking about the impact of BVD, here we have our little USS beef producer ship sailing through these waters. And the tip of that iceberg is the classic PI. And we hear a lot of, well, I tested and I didn't have PI, so I must not have BVD. There's a lot of BVD, the associated disease and loss, to generate one PI. To get a PI to hit the ground and live, you kind of have to have an alignment of the planets. It has to be infected at the correct time in pregnancy. It has to live. So there has to be a lot of acute infections walking through that herd to get a PI. Don't just think that, oh, I had one PI, so I had one infected animal. You had a lot of infection to get one PI. So let's deal with question number 1. How can a calf be PI positive, but the dam is negative? Well, there are 2 ways to generate a PI. You can have a BVD be negative dam. Either she's never seen the virus or she has inadequate protection, either because she was infected with a different strain or the vaccine she didn't give her adequate protection. And the BVD field strain is able to infect her, and then she passes the virus to her calf, and you get a PI calf. The other way is you have a PI positive dam, and she's always replicating the virus. She passes it to her fetus, and the fetus then gives rise to a PI calf. a PI dam always gives rise to a PI calf. There's no way that a PI dam can't not have a PI calf. But most of the PI calves that we find in the field come from this route, when a negative dam gets infected, and a lot of those are first calf heifers. So the timeline for BVD infection, and this is what I said about all the planets have to align to get the PI to hit the ground. There's only a narrow window where that PI can happen. It's 60 to 120 days that you get this animal who's born that can't tell the difference between the virus and itself. That's what we mean by immunotolerance. So before then, you get early embryonic death, reabsorption. You can get abortions with BVD, but you tend to get more just delayed breeding. If they're infected after this window, you tend to get more abortions, stillbirth, weak calves, and birth defects. And then there's this window where those animals are infected in utero. They're born with antibodies to BVDV, indicating that they were infected, but they fought it off. And for a long time, we thought these animals were home free, that they were okay. And what we're beginning to realize as we look at the records for these animals is that they have a higher incidence of disease and lower reproductive rates. So next question, I've been vaccinating my herd with a killed 9-way vaccines. How did this not protect my cows from delivering a PI calf? So why do BVD vaccination programs fail? Well, notice I don't say why do BVDV vaccinations fail, I say vaccination programs fail. Because the vaccines all have been licensed, they have been tested, they do reduce the incidence of persistent infection. Now, then that's what it says on the label. If you notice, the label doesn't say it absolutely 100% prevents persistent infection. It aids in the reduction of. No vaccine is 100%. And it's even more difficult for BVD vaccines to be 100% than other vaccines to be 100%, because it's a higher level of protection that's needed. The requirement for a typical vaccine is just prevention of clinical disease, so that the animal gets infected, but they don't get sick. So like when you get the flu shot doesn't mean that the flu virus doesn't replicate in you at all, that it doesn't infect you. What it means is the flu vaccine, when it infects you, won't make you as sick. And that's what we ask of a typical vaccine. But the requirement for fetal protection that we're asking from a BVDV vaccine means that it prevents the passage of the virus from the dam to the fetus. And that means we almost need to have what we call sterilizing immunity. That means no viral replication at all. And that's really hard to achieve. We know that when we use typical BVD vaccines, we're able to get a 1:16 titer that refers to the amount of antibody in the blood after vaccination. And for licensing, they have to have at least a 1:16, and the vaccines do hit that, or they would not be licensed. But we know from other studies that to prevent sterilizing or to promote sterilizing immunity, we need titers of 1 to 256, which means we need many times higher protection to prevent fetal infection. So it's a tough bar. It's a high bar to hit. And so what gets in the way of this strong immune response? Well, you need time for the immune response to develop. Once you're vaccinated, it takes 10 to 14 days minimum before you have enough immune protection. And that's the time it takes your immune system, or the cow's immune system, to recognize that pathogen, to recognize the virus or the bacterium, and to make the necessary antibodies and cells to fight it off. So it takes a while to set that up. The other thing that might happen is the challenge occurs after the duration of protection has expired. What I mean by that long phrase is it's too long after vaccination that you expose them to the virus. So there are very few vaccines that are a lifetime vaccine, particularly in the veterinary world. You have to keep boosting that year after year. And if you are too long after a vaccination, the protection wanes, it decreases over time. Now, the other thing that can happen is antigenic variation between the vaccine and field strains. What I mean by that is the vaccine strain and the field strain are just too different. What the protection that you generated with the vaccine doesn't match the field strain. And then we'll talk a little bit more about that later. The animal is not able to mount a good immune response, and everybody wants to throw rocks at the vaccine up here. You know, it must be something wrong with the vaccine. But frequently, it's the way we present the vaccine to the animal and when we present it to the animal, because animals need to be healthy and in a good state to respond adequately to vaccination. So things that get in the way of that are stress. If they're sick, they're already infected with other viruses or bacteria. And then if they're immune suppressed, there's a genetic component to this. Some animals just don't have a very good immune system. Others, because of what they were going through at the time, their immune system's not able to respond. And that's called immune suppression. So what I'm showing you here is vaccination in the bell curve. And when we talk about the bell curve in any biological event, when you look at the result, it looks like a bell curve. So if we vaccinate animals, and this is the number of animals, and this is the vaccination response, and the higher this curve grows is the better the response is. This response is better this way. This is the greater number of animals in that group. In any vaccination scenario, not everybody's going to respond the same way. There's going to be some animals that don't have a very good response for one reason or the other. Maybe they're not able to have a good response. Maybe they're stressed out. Maybe they have another bacteria or virus. For some reason, they're not able to respond very well. And then we have these other super responders at this end who respond very, very well. Now, you might say, well, why don't we just breed super responders? The problem with super responders is sometimes they're not very good at growing or doing other things because they're using so much of their energy in a physiological response. So kind of think of people with asthma have an extreme immune response. You really don't want to live up the high end of the curve here. You really want to be in the middle here. And if we drop a line and say, this is what we need for a level of protection, you're going to see that some animals are going to fall below the level needed. And the greater the level needed, if this would go this way, remember here's our vaccine response, the greater the level of response needed, the more animals we're going to see on the wrong side of the curve. So we have two things going here with BVD. One is that we can't really afford to have any PIs, reducing your PI numbers by 10% and still leaving 90% of them there isn't adequate to stop the disease. The other is that it takes such a strong immune response to get protection that our line's probably right here. So we need a strong protection, and it's difficult to achieve. So, remember I said I'd come back, circle around back to viral variation. Well, now I'm going to tell you a little bit about viral variation and why sometimes we have to add both a BVD1 and a BVD2 to a vaccine to get them to work better. And we're thinking maybe we even need to add more BVDs to them. And so that first sentence, elimination of virus by immune system is dependent on the recognition of viral epitopes. Viral epitopes are little pieces of the virus's coat that the immune system can latch onto, and that's where they recognize that that is a virus, and that's what the antibodies stick to, and the immune system uses that to tell itself that this is what they need to attack. Now, the viruses that cause BVD belong to different groups. Those different groups are called species. Remember, I told you that slide of BVD1 and BVD2 and Hobie-like viruses. Those are 3 different species of virus. And within those species, there's different subgenotypes. We know that the species and subgenotypes can have different epitopes, so that if you have a good immune response against BVD1, your immune response against BVD2 might not be good enough to protect against BVD2. So these differences in epitopes between vaccine viruses and field strains can lead to reduced protection. So question number 3. Now that I have BVD in my herd, what's the most appropriate vaccination protocol to use to protect my herd against developing another PI BVD calf? Well, let's talk about your approach. Management of a BVDV positive herd. Vaccination may not be the place to start. First place to start is detection and eliminate all PIs. Because if you do not eliminate all PIs, any vaccination program is going to be hamstrung. In a contest between PIs infecting animals and vaccines trying to prevent infection, the PIs are going to eventually win. And remember, they don't have to win very often to give you very many PIs. And only a few PIs can cause you some huge problems. So really, you need to start here with that herd. So the first question to ask once you have a PI identified in your herd is not what vaccine I should use. Your first question is, did I get rid of all the PIs? The next question is biosecurity. You need to adopt some management practice that reduce the entry of BVD into your herds. You need to kind of be a little bit of a detective and try to figure out how that virus got in and see if you can plug that hole so it doesn't get in there again. And then we come to vaccination. So remember that our immune protection needs to be established before exposure takes place, a 2-week minimum. So if you're going to protect that herd now, some of your herd will have been exposed to that PI, but as you've gotten rid of the PI and your next generation of calves, you need to remember to protect the vulnerable. And the vulnerable animals you need to protect is that fetus. And the best way to protect the fetus is to have a strong immune response in the dam. So you want to make sure that prior to breeding, they have an immune response to the virus. So a vaccine prior to breeding is a great way to go. Animals entering new production units, if you're moving them to a feed yard or you're to a calf grower, or if you're milking to a milking herd, those animals are going to be challenged with new viruses, particularly more BVDs. They need to be protected. And then young calves who are protected by colostrum when they are still nursing, as that colostrum protection wanes with age, they aren't protected anymore. And so you have to provide some type of protection for young calves in that period between when they have colostral protection and when they're going to be challenged again. So the question then is, when can you vaccinate a calf? And that is a good question. We have some data that suggests that vaccination using killed vaccines in calves that are still nursing and have colostrum may not be too effective. Modified lives are somewhat effective. A number of producers and veterinarians have gotten together and worked out different scenarios for vaccination. Most of them would suggest some type of calfhood vaccination and then another one before breeding. But that is something you really need to sit down and work out with your producer, with your veterinarian. But what your goal here through all this is to provide for a lifetime of protection for the animal, from the time they were a fetus to the time that they are a young calf, to when they're first enter a breeding herd. or if they're going to go into meat, then as they enter the feedlot, that they are protected at all times. And remember that that protection has to be in place before they need it. So the day that they see the virus is not the day to be vaccinated. You need to have that in place, ideally 2 weeks before they see the virus. So question 3.1, this is kind of a continuation of question 3. My question is, how long does a field remain contaminated after all PI steers are dead? That's a good question. And to answer it, I need to tell you a few things. One is, how do infections happen? And animals get infected with BVD by direct or indirect contact with mucous membranes. They don't get infected through dry skin. It has to come through the nose, mouth, or the genital tract. Or if they ingest something, it can come through the digestive tract, but primarily it's nose, mouth, and genital tract. The virus doesn't seem to aerosolize, and what I mean by aerosolize, it doesn't fly around on the breeze. It's almost a direct contact has to be made. So the sources of infections are large droplets expelled during breathing. So if you've ever looked at your cattle in the morning when it's cold and you see this cloud of vapor coming out of their mouths, it's that cloud of vapor that's infectious for other animals. Other sources are aborted materials that are on the ground. Animals will nose around in them and eat them. Any fluid that comes out of a PI animal or a transiently infected, but not at the same level, such as tears, urine, feces, are infectious, will contain virus. Contaminated colostrum in beef operations, you probably don't do this as often, but I see quite often in dairy that they freeze down colostrum, and if they don't check that colostrum, they may have frozen down colostrum that had BVD in it. And then there's this question, and I have a lot of question marks because we have anecdotes, stories that people tell me, but I don't have really hard data generated under controlled conditions. So fomites, and what we mean by fomites are dusts and bedding and things like that might be in penning. Hair, the reason I put hair down there is when hair is pulled out, it usually comes out with the hair follicle, and there's enough virus in the hair follicle that you can actually detect persistent infection by looking at hair. Salt blocks. And the reason I have salt blocks comes back to the next one, wildlife. We have wildlife that gets infected from cattle. And we are still trying to determine if wildlife spreads it back to cattle, but we wonder whether they come in contact with the virus through salt blocks or when they get into shared feed and water. So I have question marks here because I don't have good data that tells me how long it can hang around in there. What I can tell you is this. The conditions that are conducive to BVD hanging around are cold, wet, and dark. So the virus does real well in a barn in the middle of winter. It will hang around for a while. Conditions that are not conducive to BVD hanging around are hot, dry, and sunshine. So, and I'm not saying you can't get infected with BVD in a hot, dry climate that has a lot of sun. What I'm saying is when materials dry out and when they're hit by the sun, the virus in them tends to be destroyed. When you don't have sun hitting it, when it tends to be cold, and remember, the way I store virus after I've isolated it, I just put it in the freezer. So freezing doesn't hurt the virus at all. In fact, it preserves it. It likes to stay wet. And once again, if you keeps it away from sunlight, it's going to hang in there a while. So it is relatively easy to clean up. You can just use like Dawn dishwashing detergent. Will kill the virus. You don't have to go to extreme lengths. The problem is that the places that we house cattle have a lot of porous surfaces. So it's hard to clean all the way to the bottom of all surfaces. So What I can tell you is I don't know how long that field will remain contaminated. Certainly, we get more infection from cow to cow, from steer to steer than we get through housing. But we do know in feedlots, it seems that if you've had a lot of PIs in one pen and you put clean animals in that pen after the PI's out, you tend to have some BVD running through again. So clean up as much as you can, get rid of dirty bedding, pay attention to boots and shovels and trailers and all that to get them clean as you can. So question number 4, what vaccination is the best to use to protect calves that have been purchased from a livestock market and exposed to a PI calf while in the market? And secondary to that, what about calves purchased off the farm? Well, remember we talked about it takes at least 2 weeks to develop a protective immune response. If that's animals already been exposed to a PI and infected by a PI, vaccination is of little value at that point to that animal who's already infected. So we do a lot of vaccination, however, of animals as they come off the truck, as they come to your production unit from the feedlot. Is that a worthless thing to do? Well, from a control standpoint, it's certainly not an optimum time to vaccinate, but there may be some value, because if you look at it from a group perspective rather than just as the individual who's already been infected, maybe not all individuals have been infected in the group. Some of them will be infected by animals who have been infected. And so if you do get vaccine into them, you might be able to slow down or limit that infection that occurs in animals that are acutely infected by the PI. So, and what vaccine would be best? I can't say that one company's vaccine is better than another company's vaccine. I'll let them throw that data out at you. What I can say is all vaccines are under the same guidelines for licensure, and they've all passed the same guidelines that they need for licensure. I can tell you that in a situation where you know that there has been an exposure and you want to limit it, you need to get the immune response going as fast as you can, and modified live vaccines deliver a faster protection than the killed vaccines, which require 2 doses. So there you're already out to at least 4 weeks, whereas with the modified live would be 2 weeks. But once again, I am not recommending or saying that vaccinating in the face of exposure to a PI is going to make all your problems go away. It's not. It may limit them somewhat, but you still have that exposure that preceded the vaccination, can't be stopped by a vaccination. So question number 5, I test and pull out PIs immediately. Seems like the damage has already been done to the other calves. My main concern is what should I do with the calves that have been exposed? So that's kind of continues with what we've been talking about. So let's talk about life after the PI's been removed. Factors affecting herd recovery are the acute transient infections may still be circulating. Even after the PI is gone, it's still from one acutely infected animal infecting another animal acutely. And the length of when that's going to happen or how long that's going to last goes back to how mean a virus it is, how tough a virus. Not all BVD viruses are equal. Some of them are a lot harder on animals than others, and those that are really hard on the animals tend to be shed at a higher rate and for a longer period of time. And then if you happen to have another virus or a bacteria going through the herd at the same time, BVD is going to hang around longer. And the age and maturity of the immune system. What I mean there is that an older animal might be equipped to fight off the virus faster than a young animal, particularly a young animal that hasn't received adequate colostrum. Now, herd may be slow to recover optimum production after exposure to multiple PIs or PIs with different carrying different BVD strains. So if you have a situation where you have, say, instead of 1 PI in a group of 30, you have 5 or 6 PIs in a group of 30, those animals are going to have a harder time coming back. If those 5 in that group of 30 have 2 or 3 or 4 different strains that they're carrying, each one carrying a different strain, that's going to be harder on the group too and take them longer to recover. And then let's talk a little bit about the effect on young calves, on feeder calves. And contrary to what Adele sings to you, what doesn't kill you does not necessarily make you stronger. So I'll talk a little bit about BVD in young calves. And I'm going to throw a little terminology at you here. The immune system of a calf when it hits the ground is not mature. It's not set up. It's not what you see in an adult animal. And so there's a relationship between the age of the cattle and their susceptibility to infection. That's why a lot of diarrheal diseases are much more prevalent in young animals than old, because young animals just don't have the immune capability of fighting them off. And so we know that there's kind of a maturation process that's going on in the immune system on that calf from between the time it hits the ground until maybe the first 6 or 7 months of life, primarily. And one of the organs that figures out to a large degree is the thymus. And the thymus is very large in the newborn, and as the animal ages, it gets smaller and smaller. And the reason the thymus is big in the newborn and smaller in the older animals, the thymus is being used to set up the rest of the immune system. It's directing what's going on and telling the immune system how to become a mature immune system. So when you have stress and disease, you get this thymus getting small faster than it should have. So it starts to disappear before the immune system's completely done with it. So what I'd like to show you here is what happens to a thymus in calves that have been exposed to BVD. So in a non-infected animal, this is the weight of the thymus, and that bar here just shows you the variation. So in a healthy calf, good colostrum, hasn't been exposed to anything, you'll have a good-sized thymus. If it's been exposed to just a typical virulence BVD, which may not even give you any clinical signs that you're going to note, no diarrhea, no respiratory disease, maybe a temp for a day, might not even be high enough that you notice the animal's depressed. We have less than half, maybe about a third of that thymus left. If we go with a virus that's a tougher, more virulent type of BVD, we'll have even less of the thymus left. And what we think is happening then is we're actually losing this thymus material that the animal needs to set up a healthy immune system. So not only when we talk about providing a healthy immune system for that animal for life, it's not just getting vaccine into them, it's keeping them from getting infected with things like BVD, which hits the immune system before it's ready to defend itself completely. Not only can it not fight off BVD as well as a mature immune system, BVD actually ends up messing up the immune system, and that messed up immune system could affect the animal for life. The other prolonged effect of infection we see when we get these severe infections with BVD in young calves, where they have a really diarrhea, might be a bloody diarrhea, but it's definitely a very watery, severe diarrhea. And we were looking at these animals, they'd all cleared virus by 14 days post-inoculation, post-exposure to that virus. And the study we did then just looked at their growth rate, and we went 60 days out. And this is at 30 days post-infection. They've all cleared the virus. None of these are PI animals. So these are the ones that were infected up here, non-infected down here. Now, a calf raiser getting these guys in would say, well, let's test them for PI. They don't look very good. Maybe bad genetics, certainly poor nutrition. Something's going on with this lot of calves. And this lot of calves, well, that's a good looking lot of calves, but this is only because they were infected with BVD. These are same type of calves, housed under the same conditions, fed the same way. We only kept these guys for 60 days. They did not catch up to the non-infected. Perhaps at some point they would have, but certainly for the first 30 to 60 days, they still look pretty bad. So BVD, we say, I didn't find a PI, I didn't have a BVD problem, I just got kind of skunky looking calves. It could be a BVD problem. It could have been an acute infection. The acute infection's already been eliminated, but the damage done to these young calves hasn't been recovered yet. So question number 6, herds with resident PIs have high titers, so why isn't leaving a PI in place an effective approach to achieving herd immunity? In other words, isn't a PI, because he infects everybody, isn't that as good as a vaccination system? Well, the problem with that is living with the PI is the equivalent of being vaccinated every day. Yeah, they are good at getting other animals infected, but when you have a PI there, it's like getting your flu shot every day. It's like getting that shingle shot every day. Imagine how that'd make you feel. Think about how your animal feels when they're constantly fighting off the virus being shed by the PI animal, because when you mount an immune response to a virus, you eliminate it from your body. That doesn't mean you quit responding to it. That means you just respond to it stronger the next time you see it. Well, if you're seeing it 7 days a week, 24 hours a day, you're going to be expending a lot of energy in fighting off that virus that you would have been using for growth and reproduction. And also you're calling up some non-helpful immune responses because frankly, you're just having that virus being presented to it day after day after day because it's being shed by the persistently infected animal. You're poking that immune system with a stick day after day after day, and you're pissing it off. And after a while, the immune system starts giving you some non-helpful immune responses like inflammation. So think about having an asthma attack. That's a non-helpful immune response. That's a pissed off immune system that's angry and doesn't know what else to do because it keeps getting presented with the antigen that makes it respond. So that's why we don't want to keep a PI there. Question number 7. So hopefully I've convinced you that getting rid of PIs is a good idea. So let's talk a little bit about detection. Can I pull my ear notches? How sensitive is a snap test if I pull samples? Well, once again, just like with vaccination, detection for BVDs has an added level of difficulty than we see with detection to any other disease. So for detecting outbreaks of other pathogens, you just have to demonstrate that the virus is present to say that the herd is infected. You don't need to identify every infected animal. You just have to say it's there. When detecting PIs, you have to identify every single PI and get rid of them. So it requires a very high level of, very high level of sensitivity and specificity of your test. So let's talk a little bit about detection targets, just so you know what you're getting when you're getting a test. When you get an antibody test, and it will be expressed as a titer, either 1 over a number like 1 over 16, or they may just say you have a titer of 16. That really is actually 1 over 16. That's just antibodies against BVD, and that doesn't tell you whether the virus is still there or not. It just says that the virus at some point was there, that the animal was exposed at some point. It could be a vaccine, it could be field strain. We can't really tell by antibodies for sure, but those 2 tests will be a serum neutralization or an antibody ELISA. So when you do a test, if you're just sending in serum, you're probably just getting an antibody test. For the detection of whether the virus is actually there, for replicating viruses, be virus isolation. Most of you are not going to send off samples for virus isolation because it takes a long time and it's really relatively expensive. Probably what you're going to be doing is looking for tests that are looking at viral proteins or viral genomic material. So the specific target-based methods that are commonly in use now is the genomic target, which is PCR, which stands for polymerase chain reaction. And what that is just a test that amplifies the genetic material, the virus there, detects it, amplifies it, and so they can see it. So if there's a little bit of the DNA, RNA of whatever pathogen you're looking at, the polymerase chain reaction is a way of detecting it. Now, viruses are made, there's a protein coat on the outside and the genetic material on the inside. PCR detects the genetic material. Antigen targets detect the protein coat on the outside. So immunohistochemistry, when you send in an ear notch and they fix it and they look at those sections under a microscope, that's immunohistochemistry. That needs to be done by a pathologist, and you'll get a reading on those. Or antigen capture ELISA, where they leach the protein out of the sample, like if they soak an ear notch, the protein comes out, and then they use antibodies to capture it, and then they get a reaction that way. That's whether the protein's there. And the SNAP test that you talk about, that's actually just looking for virus protein. And it's actually, in a way, uses the same technology as antigen capture ELISA. So the next question then is to pool or not to pool. You can pool samples. It is possible to pool samples for both the PCR-based test and the antigen-based test. The question is, should you? Now, it may be cheaper initially, but it may be penny wise and pound foolish. There's the initial cost versus the timely identification and accuracy. So where I say it depends on whether you want to do this, the questions you ask, is it surveillance or etiology? What I mean by that are you don't have it for surveillance is you really don't think there's BVD there. You just want to check. You just want to stay on top of it. So you're just surveying. You're just asking the question, testing every year to see if it's there. What I mean by etiology is you know you have BVD losses, and you know you've had at least one PI in that herd. So you're specifically looking for it, and you need to get rid of all the PIs. So pool or not to pool, if you're just surveying and you don't think it's there, you're just trying to be careful, maybe pooling might be the way to go. But if you need to eliminate every PI and you know there are PIs there, you really don't want to pool, because you want to identify those as quickly as possible. Because if you pool and you get a positive, you still have to go back and retest again and pull them out. The type of operation, the feedlot, pooling samples, then you have to go back and figure out which animal in that feedlot, and they're being mixed together. It might be hard to do a follow-up and find them. You have to, I guess what I mean by type of operation, you have to figure to yourself, how damaging is it going to be not to find the PI? How damaging is it going to be to leave the PI there? And then if the PI status is claimed as a condition of sale, I would suggest that those can't be pooled. That if you're saying this animal is not PI, you should have an individual test on them. Now, the reason I kind of look at pooling with the jaundiced eye is my history. I developed some of the first PCR primers for BVD. We developed them in 1994 and then improved them in studies we did in 1998. In the first study, we used 140 different isolates. In the second study, we used 345 different isolates. These not only were different persistently infected animals, these were animals persistently infected with different field strains. Dr. Kennedy came along in 2005 and based on 4 ear notches from 4 different PI animals, 2 of which had the same virus, said that he could pool them and still find pool them with ear notches from negative animals and still find the virus. Well, from there, people just kind of went nuts with pooling. We saw people pooling 3 and 5 and 10, and some were even pooling 100. 25 was not unusual at one point. And what we started seeing in 2006 is a number of laboratories were able to generate reproducible results using these pooled samples. And then in about 2008, 2009, we started having antigen capture tests that were commercial tests, and followed by the SnapCap tests being marketed directly to private labs, not diagnostic lab, and the SnapCap directly to producers. If you look on the literature that comes with those tests, they are not licensed for pooled samples. They're licensed for individual samples. If you choose to pool samples with those tests, the manufacturer doesn't guarantee that they'll still work. And that's because the manufacturer has designed those tests to be used for individual samples. When you pool samples, you dilute. And by diluting, you water it out. So why would that be important? Well, that's important because not all PIs are the same. This goes back to that question of why do some PIs look really good and some PIs dwindle and die? Why do some PIs make it to market weight and some PIs don't? And an unfortunate case for South Dakota State University, but a case that was great for science in general. South Dakota State bought 130 pregnant heifers. They were trying to establish a new breeding herd for the university there. And in the process of buying those heifers, got them all exposed to BVD. And they consequently had 44 persistently infected animals hit the ground who were infected at about the same time and who are all infected with the same isolate. And what we were able to see at that point is shown in this pie chart. So we had about 41% of those die from mucosal disease. I haven't talked at all about mucosal disease tonight, but that's a disease that almost only happens to persistently infected animals. It's a really ugly hemorrhagic type of disease. About 15% of them, sudden death, no lesion. The 27% made it to market weight, sold for slaughter. And about 12% died at neonate, and about 5% just were poor doers, didn't thrive, and were eventually euthanized. So in this group, there were animals that looked completely fine, and there were animals that looked pretty scrubby, and then there was everything in between. Well, in addition to looking different and having different health outcomes, they also have different shed rates of virus and different amounts of virus in their tissues. So they all had virus in their tissues. They all were persistently infected. They all were positive by ear notches, but they didn't all have the same amount of virus in each ear notch. And this was revealed in a study we did. We took about 200 ear notches from different animals. And then we determined how much virus was in them, and the virus was in them we express as tissue culture infected doses per mil of solution that we soak the ear notch in. So we'd soak the ear notch in a mil of solution, and then we'd see how much virus leached out into the solution. And so what you see here is you had some of those animals had over 10,000 viruses per mil leach out of their ear. It's a lot of virus. Most of them were somewhere in here between 100 and 1,000 viral particles per mil coming out. But then there's this group that had just 10 viral particles coming out. Now, your detection methods start kind of falling apart around here. So if we dilute these animals you could dilute with 1,000 negative animals. You'd still be able to get them to be positive. You could still detect them even if they've been diluted with samples from 1,000 negative animals. These guys who don't have a lot of virus, if you dilute them 1 to 2, 1 to 3, 1 to 5, 1 to 10, you may no longer be able to find them. Now, the next question is, well, they're not carrying much virus in their ear notch tissue, therefore they're not shedding too much. But we didn't see that correlation. These animals may not have much virus in the ear notch, but they certainly were still shedding virus and were infectious. So missing these guys is not an inconsequential thing. And that's why I don't like pooling, because I think you start to miss these guys at the bottom. And I don't think you can afford to. And then but we didn't find a PI. Well, the problems you may be having with BVD might not be the result of a PI there. It might be the circulation of acute infection among naive animals. So multiple herds rotating through a facility with regular additions of animals with BVD there. You don't have to have a PI. Just the acute infection that's hanging around for 2 weeks to a month or so is enough with that constant rotation of animals to just keep it going without a PI there. It works better with a PI, but it can happen with just acute infections. And then the PI maybe isn't in your herd or your feed yard. The PI contact may have been in the sale barn, not necessarily the animal you bought, but the animal that it was in contact with. The use of bull raisers, contact with non-bovine populations. Remember, wildlife can carry BVD. So can milking goats. So can sheep. Llamas, alpacas can all be persistently infected. Use of contaminated bovine products, colostrum. Semen can have BVD in, especially if they're using extenders that are made from cattle products. And fetal calf serum, you say, I never use fetal calf serum. Well, if you're doing embryo transplant, you might. That can be contaminated. Cattle shows. You can bring BVD home from a cattle show. And just because an animal is persistently infected doesn't mean it's an ugly animal. A certain percentage of them are stumpy, bad-looking animals with poor hair coat. Some of them aren't. There have been 3 generations of scientists studying BVD at the NADC. The first one was Arlen McClurkin. And Dr. McClurkin did a lot of the groundbreaking work. I inherited Arlen's office when he retired, and a picture that he left me on the wall was a picture of a bull that won purple ribbon at the Wisconsin State Fair that was persistently infected. It was a beautiful bull, but it was a PI. And then the other thing is you might be having problems with your test strategy in that you are not testing everything. So when you're trying to find the PI, you don't have to test everything every year, but that first year you may have to test all the calves and test any cow that doesn't give you a calf. You can assume that if a cow gave you a normal calf, a non-PA calf, that cow is not a PI, because remember, PI calves always give rise to PI calves. But also, so you can start by testing the calves. Any PI calf could possibly have a PI dam. You have to go back and test that PI dam. You also have to go back and test anything that didn't give you a calf. A cow that failed to breed that year might be a PI. So your bull might be a PI. So your testing strategy, and this is something you need to sit down. It's not something you can write on the back of a napkin in 5 seconds. You need to sit down and really think about it and talk to your veterinarian. Spend some time, figure out what your strategy is for testing. So back to the trenches, back to these original questions we had. How long will it take before you see sickness because of those PIs were there? Now remember, it really depends on how bad the virus was, depends on what else is going through that herd, depends on how much stress is there on those animals. So that's pretty variable. Usually it's going to be within the first 2 weeks, but it could be longer than that. How long after infection before the immune system, it affects the immune system of the calf, and how long will the immunosuppression last? Well, it's going to start hitting the immune system within 3 to 4 days of infection. How long that immunosuppression is going to last really depends on, once again, what viruses they've been infected with and how healthy the animal is. What else is going on? So it could resolve itself in a couple of weeks. It could last for a while. What causes a difference in severity of transient infection cases in BVD? Once again, type of virus you got in there, whether it's one of the mean BVDs or one of the mild BVDs, whether those animals are under a lot of stress, whether they've been vaccinated well. You've got the whole range of those things going on. When can one reasonably expect the virus to be out of a group of calves? Or do they've developed protective immunity so you can safely merge them with other calves on the farm? Boy, isn't that a good question? And isn't that a crapshoot? Depends on who you're going to merge them with. Keep them separate as long as you can. Minimum 2 weeks, 30 days would be much better. And if you're going to mix them with pregnant animals, man, that is so dangerous. So yeah, as long as you can possibly keep them separated, do. I know in all management systems that isn't possible, but as long as you can keep them from mixing with your other animals, the better. Can you shed light on transmission across fences, or if they can touch noses, transmission in water troughs and bedding? Yes, you can have transmission in bedding. I don't know about water troughs. I suspect that's true. Certainly nose to nose touching transmission, that's a great way to transmit it. As we talked about, I don't know about salt blocks. We suspect, but we don't have good data yet. And how do you clean and disinfect? Get rid of all loose materials that you can, all soiled beddings, clean any surface that you can clean, particularly trailers, shovels, boots. If you can get sunshine and light to it, it's great. But just remember that virus can hang out if it's a wet and cold situation. So if you can get it dry and warm, it's going to help. So I have used up my time. I apologize. I ran a little bit long, but if you have any other questions, I'd be happy to try and answer them. Okay, I'll just go with them in order and I'll do my best, folks. Can modified live BVD vaccines cause the animal to receive the illness? We gave our calves a modified live vaccine and lost several. The next year we did not use modified live and saw very little illness. Animals that are stressed and not feeling well otherwise may react poorly to a modified live vaccine. Remember, the BVD is not the only component in the vaccine. What we've seen is particularly IBR out of vaccines causing problems. You can't get a persistently infected animal from a vaccine, because the vaccines are cytopathic viruses, the modified lives, and cytopathic viruses can't cause persistent infection. But just like when you get a shot and it makes you feel kind of crappy for a while, imagine getting a shot when you're already not feeling well. It's going to make you feel even worse. So modified live viruses are replicating viruses. They're not going to cause the most severe form of the clinical disease, and they're not going to cause persistent infection, but they are hard on the animal, and it's kind of a pay me now, pay me later. So they're going to make the animal feel a little bad now, but down the road when they get exposed to a real live virus, they're not going to have such a hard time. But when you're vaccinating, you want them to be in the best position they can be to receive that vaccine. Because if they are stressed, if you castrate, dehorn, wean, and vaccinate all the same day, just think what a shitty day that is for an animal, and how you're giving them live virus. Even if it's attenuated and weakened virus, they're not going to do well. So really look at how you're handling those animals. The vaccines aren't dangerous. And if they did kill animals outright, they wouldn't let them be licensed. If they caused outright clinical disease, they wouldn't let them be licensed. But that doesn't mean they don't do anything to the animal. The virus replicates. Okay. Is intranasal vaccine helpful? A lot of data on there, both ways on intranasal vaccines. There's no definitive study that says that they are absolutely safe, and there's no definitive study that says they're not good, that, they do give you an immune response, but I'm a little nervous about them. But I tend to be kind of nervous about things anyhow, because in my career, I never got calls from people who were happy. I always got calls from people who were wondering who was the rat bastard they were going to sue because they just had a horrible wreck. And an intranasal vaccine in a sick animal is a bad deal. So that's intranasal in a healthy animal, probably okay. Intranasal in an animal that already has something going on, and intranasal respiratory could be a problem. But that's just
Of course, there are no intranasal BVD vaccines, right? There's
not currently.
Right, that's what I thought. They have the other viruses, but not BVD in there.
Yeah. There have been some, and they've been back off the market too, so that might tell you something. Once again, I should qualify, I am not a veterinarian. I do not have a license, and I cannot practice. So I can't tell you how to vaccinate. I can just tell you what little information I know. Okay, is there a difference in the infection contracted if a green or healthy calf is exposed to a PI calf as compared to being exposed to just a sick calf with BVD? Not okay. It won't change how mean the virus is, how tough the virus is, whether it's a PI or an acute infection that's being transmitted. What's maybe different between the being exposed to an acutely infected animal and being exposed to a PI is that the PI is probably shedding more virus. So you might have heard when they're talking about the coronavirus viral load, how sick people get depending on how much viral load they were exposed to. The viral load is usually higher with a PI animal than an acutely infected animal. So they're shedding a lot more virus. So it's a more efficient infection, and you may get a little bit sicker, but all that goes out the window when you have one of those really tough BVDs, the mean BVDs, the high virulence BVDs. Those acutely infected animals are just as bad as the PI. So, and does the thymus regulate growth? The thymus is more involved in immune regulation, but a healthy animal with a healthy immune system will grow better. So it's all connected together. It doesn't have the primary responsibility for controlling growth, but certainly a healthy thymus contributes to healthy growth. And is there an effective intranasal BVD vaccine for calves? I think Dr. Arnold just answered that. So not currently. If so, when should it be given? And really, it's not there yet. Should it be given with another intranasal vaccine, just as Enforce 3? The things we do to animals when we give them multiple vaccines at the same time, and I know from a management standpoint, you only want to handle those animals so many times, but think about you getting your smallpox shot, your influenza shot, your shingles shot, and your pneumonia shot all the same day. One, you're going to feel crappy. And two, how does your immune system know what to respond to? And the other thing about the intranasals, you're actually asking that virus to replicate in the intranasal system, but not get into the lungs. So I think you get to the point where you start overwhelming animals by giving too much at once. Okay, preferred vaccine protocol for a cow-calf herd. Well, once again, I'm not a veterinarian, but you want to make sure that you have those animals protected as a fetus. And the best way to protect the fetus is make sure the dam's protected. Best way to make sure the dam's protected is having some vaccine in there prior to breeding. And we also have to protect the calves, so it's good to have a calfhood vaccine in. And then after the first calving, at least I would say one vaccine a year. Now you can go overboard the other way. Some dairies, you're seeing them get 5 or 6 vaccines a year, which I think is probably overkill. So, but you look at those vulnerable periods. I want to protect the fetus. I want to protect the calf. I want to protect the animal that's going to go into a new production system, either a feedlot or the breeding herd. I want to protect them before they enter into that herd. I want to make sure protection's there. And then as an adult, it's keeping that protection up, but in your breeding herd, it's mainly keeping that protection up so you can protect the fetus. So if I vaccinate at weaning with a modified live and booster with the same 3 weeks later, PI test comes back negative, does this mean that my calf might test positive, say, a year later? No. Once your animal has tested negative, you really don't need to test them again. The only way you can become a PI positive is by infection when you're in utero, while you're still a fetus. You will not develop a persistent infection later in life. So once in a while, you'll go to a sale barn and you'll see animals with multiple notches out of their ears. And you don't have to do, once they've tested negative, there's negative. The only time you would test again is if they've tested positive. And Michelle and I were talking about this before, that really the standard for being persistently infected, the industry standard for declaring something persistent infection is 2 tests, 2 weeks apart. Now, the problem with that is nobody wants to hold that PI animal for an extra 2 weeks separate from everything else and test it again. But so the only time you have to do multiple tests is when you have a PI animal. So you don't, once it's negative, it's negative. If that's a true negative, you don't have to worry about it. You're home free.
Is there the potential for recombination with multiple PIs when different strains are pinned together? Recombination of the viruses into new strains?
What we see more often than a new non-cytopathic strain which takes off is a new cytopathic strain which causes mucosal disease, which starts wiping out all the persistently infected animals in the herd. So yeah, recombination can happen. We've seen even recombination happen when vaccinating persistently infected animals, recombination between the vaccine virus and the persistently infected animal. This is another reason not to get rid of PIs and not vaccinate them. So recombination isn't a real big bugaboo monster on the horizon. Most of the time, what you'll get is a cytopathic virus. They'll start killing PIs. And you might say, well, great, it kills PIs. Well, the problem is it kills them by giving them mucosal disease, which is one of the most miserable ways to die I've ever seen any animal have to go through. I mean, it's unconscionable to deliberately encourage mucosal disease. It's awful. So, yeah. But as far as recombination with multiple PIs, what we don't so much see as recombination is that the non-PI peers housed with them just have such beaten up immune systems that sometimes they even start having protein accumulate in their tissue, so they almost look like a PI. Once you separate them from all those PIs, then they start clearing up again. But it's a bad deal. It's tough on everybody to have multiple BVDs around all the time. So, but I don't think from a recombination isn't the biggest terror that we should have on that.
Just some takeaway points. These are really, really important. Correctly testing. Don't delay testing. If you're a backgrounder, if you're a stocker operator, when you buy those calves, don't delay your testing. Get it done, get your answer, Get them out, because time is not your friend. Time is your enemy. The longer they're together, the more spread. So get that done. Get that done immediately. Don't think vaccination's going to help you out right away, because it won't. Takes a minimum 2 weeks to get that immune response. And that's when the calves are healthy. They get a nice immune response. When you're buying these green calves that are just weaned, they're young, they're balling, believe me, they're not going to mount much immune response to your vaccine. So take that to heart. This is important, that you get the PIs out. And really, I hear more complaining, and rightly so, that you feel like you're stuck. You got the PI and now you have to euthanize it. You've lost all that money. The testing needs to be done at the cow-calf level. It needs to be done there before those calves ever leave the farm. So that's kind of where we need to be directing our thoughts, is we need to implement testing on the farm where they're born, because those PI calves really don't ever need to leave. And that would help us out a lot. Anything to add to that, Dr. Ridpath, before we call it a night?
Just a big amen. I agree with everything you said.
Oh yeah, so Dr. Johnson said calves can be tested. We're recommending now you wait till they're about 10 days of age before you take the ear notch. Get some, evidently there's some interference right at birth, but 10 days and after, ear notch those calves, bring them in, we can test. If you get a positive, you want to go back and test the mom. But and then we can work with you from there, and hopefully figure out where the problems are. Thank you again, and good night.
Good night.